which is asymmetric multi-epitope lipopeptide vaccine model. As you know, adjuvant development is a crucial area of research for improving efficacy, safety, and specificity of the vaccine. So there is a high demand to develop a vaccine with protective immunity against intracellular bacterial and viral pathogen. This means that we need a vaccine to induce, both cellular and humoral immune response. So there are a few number of adjuvants that have been developed for human use, but still their mechanism of action is unclear. So a structural investigation to determine the structural activity relationship of the adjuvants have a rationale for vaccine design. Lipopeptides have shown promising effects to be used as an adjuvant for subunit vaccine because of their high purity and their self-adjuvanting properties and their possibility to define their structures. So previous studies on lipopeptides have shown that lipid length, number, position, and character of the lipids and spatial orientation of the epitopes in respect with lipids are effective in the level of antibody and immune response. Also, these lipopeptides are related to a type of materials that are tendency to self-assemble into particles of different shape and characteristics, and it has already been shown that particle size, shape, and secondary structure are important for the immune response. Although great researches on lipopeptide designs have been done, we thought that by putting asymmetry in the arrangement of the epitopes, we may get a better immune response. And why we did asymmetry? Because it has been shown that the macromolecules with asymmetric arrangement of the lipid hydrophilic and hydrophobic ligands are tendency to self-assemble into orderly aggregate, which means that they can display the ligands around their surface. On the other hand, biological researchers have been shown that asymmetric arrangement of subunits of macromolecules have profound effect on their cellular interaction and signaling. So we put this asymmetric arrangement of epitope by using ovalbumin as an antigen model. So we use OVA CD8 because it's important for cellular immunity, and we put also OVA CD4, which has been shown that it's effective for development of effector and memory CD8. So we aim to synthesize a library of asymmetric vaccine candidates and investigate their particle formation to see how their physicochemical properties affect on the immune response. And we investigated the impact of lipid length and position on the particle formation and immune response. This is a library of the constructs. The construct with lipid at N terminus are constructs 6, 7, and 6 prime and 8 prime. And those that have prime contain 4 copies of OVA CD8. And the construct 8, 9, 8 prime, 9 prime has a lipid in the middle of the construct. So to making these constructs, first I synthesize the lipopeptide azide building block using a standard microwave-assisted solid-phase peptide synthesis to synthesize the peptide and then attach lipid at the N-terminus of OVA CD8, and then using orthogonal scheme, replace the azide moiety at the start of the molecule. And then, again, using the same method, I synthesize another building block with lipid at C-terminus. And then we purified and characterized using mass spectrometry and HPLC to find peptides with more than 95% purity and reasonable yield. And also I synthesized another building blocks contained OvaCD8 with alkyne functional moiety, which contain 1 or 4 copies of OvaCD8, and again I purified and characterized them to find the pure compounds. Then I did a conjugation reaction to conjugate these lipopeptide azide with peptide alkyne using a Crick reaction. So first I synthesized and optimized this reaction by changing the solvent system and changing the ratio of starting material to find a product with the highest yield and purity, and then use this condition to synthesize other constructs, asymmetric constructs, which are 6, 7, 6 prime, 7 prime, and again 8, 9, 8 prime, and 9 prime. Then we self-assemble them in PBS, which means that first dissolve in hydrochloric acid and then increase the pH to self-assemble them, and look at in TM. So it's very interesting that the construct with lipid at N-terminus and showed that with C12, we saw the spherical particle, which is number 6. By increasing the lipid length, we saw the construct with the long fibril particle shape. And but the construct with 4 copies of OvaCD8, both of them showed the fibril. This means that the strength of a hydrogen bond in these constructs are higher than the strength of hydrophobic interaction. But when we change the position of the lipid to the middle of the construct, in construct 8, 9, 8 prime, and 9 prime, we saw the mixture of spherical and short fibrils. And then we look at the topological features of construct 7 and 7 prime in using AFM, and we saw that the construct that have the CD8 dendrimer has higher packing density of the other constructs. And CD analysis showed that the construct 6 for the random coiled secondary structure, and 7, 6 prime, 7 prime, and 9 prime formed beta sheet, and 8, 9, 8 prime formed alpha helical secondary structure. So this means that the location of the lipid may affect to the secondary structure of the construct. Then we moved on the effect of in this construct on the immune system. As we used the ovalbumin as an antigen model, so we used a mouse oval model, which are OT1 and OT2. OT1 is a transgenic mice, contain CD8 T cells specific for the OvaCD8, and OT2 is a transgenic mice with CD4 T cells specific for the OvaCD4. So we separate and purify these T cells and equally mix them, adaptively transfer to naive black 6 mice, and then vaccinated these mice and look at the expansion of these T cells on the vaccinated mice during the immunization course. And then after the second boost, we look at the activity of these proliferated T cells by the CTL assay. Then we challenge the mice with tumor, with ovotumor cell line, because tumor growth inhibition is part of cell-mediated immunity. And after 3 weeks, we sacrifice the mouse and look at the cytokines for the immunogenicity test. So the results of proliferation assay after the first immunization showed that the constructs with lipid at N-terminus, which are 6, 7, and constructs contain multiple OvaCD8, which are 6 prime to 9 prime, had a higher proliferation effect on the immune system of the CD8 OT1. But When we look at the kinetic of proliferation of these T cells, we saw that construct 6 after the second boost doesn't change the level of the proliferation, but construct 7 significantly increased the proliferation of the T cells to a level with no difference with the positive control. So to relate these data with physicochemical properties, I come up with a graph and this shows that construct 6, which has a spherical shape, and construct with beta sheet secondary structures had a higher proliferation of CD8. But considering the kinetic of proliferation, construct 7 stand out among the other constructs. Then the results of CD8 for the checking the activity of these T cells have shown that again construct 6, 7 and 6 prime, 7 prime and 9 prime had a higher activity to non-significant difference with the positive control. And these constructs completely inhibited tumor growth. So again, we see the same profile as I showed in the previous slide about to relate these immune response with physicochemical properties, and constructs with spherical and beta sheet secondary structures show the highest activity and tumor growth, while the alpha helical secondary structure didn't inhibit tumor growth. Then, as an important cytokine for T helper 1 immunity, we look at the cells generating interferon gamma by interferon gamma ELISpot assay. And these results again confirm that construct 6, 7, and 7 prime had generated higher interferon gamma cytokines. And also we look at IL-2 and TNF-alpha as another important cytokine for T helper 1 immunity by CBA assay, and all of them showed that again construct 7 and 7 prime had the same profile for the higher interferon gamma production. So these are construct 7 and 7 prime. Both of them have lipid at N-terminus and long febrile beta sheet structures. And then we look at the humoral immunity by doing the ELISA test against ovalbumin protein. And these results showed that construct 6, which has a spherical particle shape, and construct with 4 copies of CD8 generated higher antibody. And also these constructs generated IL-6, which is an important cytokine for the T helper 2 immunity. Then we did a further study to see how these constructs affect on dendritic cells, and by an in vitro assay. And these results also showed that the construct 7 had a higher effect on the activation markers of the dendritic cells. So in conclusion, an efficient method was optimized and developed for the synthesis of asymmetric lipopeptide macromolecules, and physicochemical characteristics of these constructs showed that inclusion of lipid at different positions affect on the particle shape, size, and secondary structure. And the construct 7, which is this construct with 2C16 at N-terminus, formed beta sheet long fibrils with higher cell-mediated immunity and dendritic cell activation. And for humoral immunity, we showed that the construct with a small spherical particle or contain multiple copies of OvaCD8 had a higher antibody response. And overall, this novel lipopeptide designed with asymmetric arrangement of the epitope can be a breakthrough for design and development of future vaccines. And at the end, I would like to thank Professor Iswan Todd and Dr. Rachel Stephenson who supervised this project, and Professor Dennis Sulan, Dr. Simon Apte, and Dr. David Pattinson in QIMR, who designed the animal study and supervised that part of the project, and Vita and Sorania that are my co-worker in the lab, and all Todd group members who helped for doing this project. And thank you for your attention.
Okay, we have time for some questions.
Yeah. The synthesis that you described, I take it lends itself to a scale-up, that you could make it in mass?
Not in mass, it was around 3 or 4 milligrams. And we're just doing these physicochemical properties and animal study, but it's possible to work out for the scale up more. Yeah.
Some CD8 T cells need IL-2 produced by CD4 cells to maintain their activity.
That's right.
So in your constructs where you got the best tumour killing, do you think that was due to better stimulation of the CD8 cells or activation of the CD4 cells to produce the IL-2 that was needed?
For tumor protection, mostly is CD8 activity that is needed, but actually it's not just through the cell-mediated immunity, and the CD4 also that generate interferon gamma also stimulate that pathway to inhibit tumor growth. So both IL-2, but IL-2 is generated in CD8 mediated pathway. So both of them could have effect on tumor inhibition.
Yeah, but some CD8 cells are not able to make enough IL-2 to maintain their own proliferation and activity. So you often need CD4-derived IL-2 to drive a CD8 response, and I just wondered if you had looked at the IL-2 production by the CD4, the OT2 cells.
No, actually just we did CBA assay. For that one we need to do intracellular staining to see IL-2 generated by CD8 or CD4 more, but no, we didn't do that. So maybe, yeah.
Any other questions? Okay, well, thank you very much for answering.